alaikum, everybody. أنا Ahmed Babikir. I'm a assistant professor in the Division of Infectious Diseases and Department of Pathology and Laboratory Medicine at Emory University down in Atlanta, Georgia, in the United States. I apologize, I don't have a functioning camera on my laptop, but I think the important thing is that everyone can see the slides and not necessarily my face. So today I'll be talking about the role of the gastrointestinal microbiome in multi-drug resistant organization decolonization or colonization resistance. And please, if anybody has any questions, feel free to kind of unmute and interrupt me because I'm not sure I can necessarily see the chat right now. It's my first time using WebEx. Okay, so I have no financial disclosures. I don't know, can you guys see the screen uninterrupted, or is there a little thing on the top?
We can see it good, Ahmed.
Okay. Okay, so today, just a brief outline of the talk. We'll talk briefly about the GI microbiome or microbiota. We'll talk about how a healthy microbiome can protect against multi-drug resistant organism colonization. And then we'll talk about how if we restore a microbiome from an unhealthy state to a healthy state, what that can do in terms of MDRO colonization. And then hopefully, if we don't go too long, if we have time, we can touch briefly on some microbiome therapeutics that are potentially coming down the pipeline, or some categories of microbiome therapeutics outside of fecal microbiota transplantation. So we'll just kind of start off briefly with some terminology review, just so we all know that we're saying the same. When we say something, what it means. So when I say microbiota, this is just shorthand for all microbiomes present in a sample or an environment. When we say microbiome, this is the sum of the genetic content of all the microbes present. And when we talk about the GI microbiota, of course, all epithelial surfaces have a distinct microbiome or microbiota that are composed of commensal organisms. And the component of these microbiota differ from site to site, so from the skin to the oral cavity, the intestinal tract, and the reproductive tract. And even amongst contiguous sites, the microbiota differs. So the microbiota of the stomach is different from that of the small intestine, and it's different from that of the colon. And the GI tract represents one of the largest interfaces between the host and the external environment. And so it plays a large role in health and disease states. And so within the GI microbiota, we have bacteria, we also have protozoa and archaea, we have viruses, and we have fungi. And specifically, when we look at the bacterial component of the GI microbiota, these are kind of the four big classes, or the four big phyla, I should say. But specifically, the Bacteroides phyla and the Firmicutes phyla make up over 90% of the GI microbiota. The Firmicutes phylum is highly diverse and includes species in the orders of Lactobacillus, Clostridialis, and these are some of the most commonly used organisms in sort of probiotics and in other emerging microbiome therapeutics. And the Bacteroides phyla really makes up the bulk of the GI microbiota, between 5 to up to 60%, depending on the health state and the person. And so again, it plays a big role in metabolism, in colonization resistance, in vitamin production, and support of other anaerobic bacteria. And the GI microbiota, as previously alluded to, plays a large role. Some of these roles include immune system maturation and functions. And this can be demonstrated by deficiency in several immune cell types and lymphoid structures in germ-free animals. So when we raise animals in a germ-free environment and they don't have commensal cells, these mice can have specific immune deficiencies. They also play a lot of metabolic functions. Um, Colonic bacteria express carbohydrate active enzymes, which allow them to ferment complex carbohydrates. This then leads to the generation of important metabolites, such as short chain fatty acids. And these short chain fatty acids will sort of come back to time and time again, since they do play an important role. There are three predominant short chain fatty acids: propionate, butyrate, and acetate, and they're typically found in a portion of 113, or a proportion, I should say, of 113, with acetate being the most dominant short chain fatty acids. And these short chain fatty acids are absorbed by epithelial cells in the GI tract, and then they're involved in the regulation of cellular processes such as gene expression, chemotaxis, differentiation, proliferation, and apoptosis. So really play a lot of essential cellular process roles. The GI microbiota, or bacteria in the gut, also play a crucial role in the de novo synthesis of essential vitamins, which the host are incapable of producing. Lactic acid bacteria, for example, are key in the production of vitamin B12. And of course, we know that they play an important role in vitamin K, riboflavin, biotin, nicotinic acid, pyridoxine, and other vitamins, which then all have important interactions within our immune system and have been also associated with many immune-mediated diseases. But most importantly for our talk, they provide a barrier against colonization with potential pathogens. And this is termed colonization resistance. So what are some mechanisms of colonization resistance? The first one that I want to touch on is the secretion of antimicrobial products. So these include, as we said, the generation of short chain fatty acids, which can impair growth of pathogenic bacteria by affecting intracellular pH and metabolic functioning. They produce secondary bile acids, which possess antimicrobial properties. So bile acids in general are produced by the liver. They're then excreted into the intestinal tract to aid in the digestion of dietary lipids. When they reach the intestine, they're then deconjugated and converted into secondary bile acids by anaerobic taxa. These secondary bile acids are bactericidal to many bacteria, including some important pathogens such as Staphylococcus aureus, some Bacteroides species, C. diff, and Bifidobacteria and Lactobacilli. And they cause membrane disruption and subsequent leakage of cellular content. And they also produce bacteriocins. Bacteriocins are short toxic peptides, which are produced by specific bacterial species that can then inhibit the colonization and growth of other species. Outside of secretion of certain toxic products, they also play an important role in nutrition competition. And this is especially relevant for bacterial strains that belong within the same species, as they often require the same nutrients. They allow for the support of epithelial and mucous integrity. And we know this again because in experiments where mice are host under germ-free conditions, they have an extremely thin colonic mucus layer. And the thickness of this mucus layer can then be restored by exposure to certain microbes. And we know that this thick mucosal and epithelial integrity is an important barrier to pathogen invasion. And while their role in colonization resistance is still being understood, there is sort of emerging theories and data that bacteriophages play an important role in excluding specific gut bacteria. And this has sort of been shown by taking feces and filtering all the cellular content and then transplanting that. And this has been shown to cause microbial shifts in microbial communities. And so clearly it's not just the bacteria, because the bacteria get filtered out. There's other things that are sort of in there that play a role in colonization resistance. And so this is all to say that these microbial communities are well established as being critical to colonization resistance. So what happens when this normal microbiota is disrupted? This is dysbiosis. So dysbiosis means the disruption or imbalance of microbiota homeostasis. And it's typically associated with disease states. And so in a normal state, we have a normal microbiome that's resistant to colonization. And then let's say you're exposed to broad spectrum antibiotics, especially antibiotics that may have anaerobic activity. And this causes disruption of the microbiome and leaves it susceptible to colonization. Then that can allow for colonization and then dominance and proliferation of MDROs. And we know that then the GI tract can then act as a reservoir for antibiotic-resistant pathogens. And colonization of the GI tract, whether detected or undetected, frequently precedes invasive clinical infection. And one example is of this population-based study in Sweden, where they took 5,513 patients that were known to be colonized with ESBL in the gut, and above 1,700 or 17,000 patients that were colonized with ESBL in the urine. And they then matched them to a healthy population that had been screened. And this healthy population were about 45,000 individuals. They then followed these three cohorts for 6 years and calculated the incidence of ESBL bloodstream infections. And you can see here nicely the cumulative incidence of ESBL bloodstream infections differed across the three groups, with only 0.02% of those in the healthy uncolonized having an invasive ESBL bloodstream infection, compared to 3.8% in the urine group and 1.6% in the stool group. And you can see the adjusted hazard ratios there are very large. And they even did downstream adjustment for age, sex, county of residence, and Charlson comorbidity score. And you can still see quite a high um, adjusted hazard rate. And so ultimately, colonization within the GI tract, as I hope I've just showed you, can then lead to invasive disease. But not only does it lead to subsequent invasive disease, but these MDROs can then be passed on to other people, either within communities or within hospitalized patients, and then also lead to invasive infection within them. So microbiome restoration is a promising avenue for decolonization, and this decolonization can then lead to, hopefully, decrease subsequent infection and transmission events. So this brings us on to microbial therapeutics, which is the administration of viable microbes to a recipient to improve a disrupted microbiome state. And typically, at least now, when people talk about microbial therapeutics, for the most part, we're talking about fecal microbiota transplantation, or FMT. And this is the process of transplanting stool from a healthy donor to a patient. And it's one of the most straightforward ways to manipulate the gut microbiota. And so if we just kind of go back to our initial diagram, here we have a disrupted microbiota, which is colonized by and dominated by MDROs. And a fecal transplant can then restore our normal microbiome and thus restore our resistance to MDRO decolonization and also lead to modulation of the immune response, restoration of short-chain fatty acid metabolism, and reinstate phage pressure as some of the sort of mechanisms that we believe lead to the restoration of a normal microbiota. And post-FMT, we see here that in pre-FMT, the microbiota will look disrupted, and in post-FMT, the microbiota will then look similar to that of a healthy donor, and this suggests that gut microbiota has been restored. So the process of FMT involves installation of stool, which is usually filtered and then diluted with saline. And we take this stool from a screened healthy donor. The donor usually gets sort of a panel of testing, which includes testing for GI pathogens, but also sort of any latent infections, especially viral infections, and MDRO, and of course, bacterial MDROs. After the mixing and the blending and the filtration and the dilution of the stool in saline, it is then instilled either via endoscopy or via oral capsules are the two most common methods. And at least at Emory, we've been calling them crapsules as a sort of inside joke. The other methods that people use to instill FMT are through NG tube and through enemas. And in particular, I'm sure many of you are familiar with FMT because it's best understood in the setting of recurrent C. diff infection. And this was sort of a landmark trial back in 2013, which studied this in a randomized controlled fashion. And since then, there have been a number of clinical trials that have been conducted, which has shown that FMT has a high level of efficacy for recurrent C. diff infection, above 90% efficacy, with resolution rates as high as 92%. Most of these studies have compared it to what was standard of therapy back then. So most of them have been compared to PO vancomycin. And because of all of this data, it has now become an important cornerstone of the treatment of recurrent C. diff infection and is included in major society guidelines, including those produced by the Infectious Disease Society of America and a number of European professional societies. So through FMT, we've seen, I should say, through recurrent C. diff, we've seen the success of FMT, and this provides a strong evidence for the causality and the effectiveness of microbiota therapeutics, and has then spurred efforts to test FMT for other indications. So with increasing use of FMT for recurrent C. diff, the loss of MDRO colonization has been recognized as a collateral benefit in these patients. And so, the first sort of studies of FMT for MDRO colonization were spurred by us seeing that these C. diff patients who happened to be MDRO colonized were no longer MDRO colonized. And since then, this has been described in an increasing number of case reports and case series. So just kind of one example of one of these is in 20 patients that had recurrent C. diff infection at a single center were administered FMT from 3 healthy donors. This led to clinical cure, so the resolution of recurrent C. diff in these patients. But these patients also had shotgun metagenomics done, and specifically the authors were looking at the AMR gene content. And you can see here, before FMT, this is the AMR gene content, which has been quantified. And you can see compared to donors, the healthy donors, this is quite high. And then you could see the reduction post-FMT of the AMR gene content. And you can see now it sort of mirrors the donor AMR gene content. And so when looking at studies specifically that are looking at MDR decolonization as an outcome, one meta-analysis included 21 studies with a total of 151 patients and found a rate of about 38 to 88% MDRO elimination. And what's interesting also is FMT can not only lead to MDRO decolonization, but can also interrupt non-C. difficile recurrent infection. So this was, I think, an interesting case report that was published by a medical student here at Emory. And this is an elderly lady who had recurrent UTIs with increasingly more resistant pathogens. And so you can see here, this is a timeline. So you can see she has an MDRO, just use it when you're, you see it starts off in 2013 and she has an MDRO E. coli. Quickly thereafter has another one. There a period of time lapsed, and then she has another MDRO E. coli UTI. And then you could see there's just sort of multiple recurrences with different MDRO organisms, including MDR Klebsiella, MDR Morganella, MDR Klebsiella oxytoca, and of course the E. coli. She then receives FMT here for recurrent C. diff. And then you could see that she was followed up for quite a long period of time, up until 2017, where she no longer had any more MDRO UTIs. A similar observation was reported in a case control study out of Mayo of 8 patients that had recurrent UTIs. They also received FMT for their recurrent C. diff infections, and they were matched to 8 patients that had recurrent C. diff, but no FMT. And you can see here, for the most part, and sorry, I should say that these patients also had recurrent UTIs. And you can see for the most part that there was a decrease in the number of UTIs per year for the FMT group compared to the non-FMT group. And specifically, the decrease was from about a median of 4 recurrent UTIs per year to 1 per year post-FMT. But FMT not only can reduce infections, but also in some studies has been shown to have a reduction in all-cause mortality and bloodstream infections. So this was a prospective cohort study of 290 inpatients, again with recurrent C. diff, since this is the only approved indication for FMT, 109 who received FMT, and 181 that did not receive FMT. And in this prospective study, the primary outcome was bloodstream infection in the 90 days post-FMT. And a secondary outcome was 90-day survival and length of stay. These patients were matched through propensity matching, and so we lose some of these we lose some of these patients, and we're left with 57 propensity matched patients. And you can see that there was only 5 bloodstream infections in the FMT group compared to 40 bloodstream infections in the non-FMT group, and this comes out to a 23 percentage point reduction for BSI and a 32% point increase in 90-day survival from 39% to 75% in the FMT group. And I think this is really nicely illustrated in the Kaplan-Meier curve, where you can see clearly that the two groups separate, and there's no overlap in the 95% confidence interval. And so this brings us to our own experience with FMT at Emory. So this was a clinical trial that was conducted specifically by Michael Woodworth, also a member of the infectious disease faculty in our division, which I was fortunate enough to participate in. And it's called PREMIX. It's a randomized controlled trial of FMT for eradication of intestinal MDRO colonization after infection, specifically in renal transplant patients. So we had 11 renal transplant patients who were then randomized to either FMT or to an enema. And so we had 6 in the FMT group and 4 in the enema group. And patients were then followed via blood and stool, but specifically via stool, for MDRO decolonization. So all of these patients were renal transplant patients that had an MDRO infection. So that's how they were initially found. They were then screened for MDROs in the stool, and only then could you be eligible to enter the trial. They were then observed for a period, and then were eligible to receive up to 2 FMT cycles. So if you underwent an FMT cycle and were still colonized by the end of the observation period, which was 36 days, So you see 6 here that were MDRO colonized, they went through the FMT cycle, and if they were still colonized, they were then eligible for another FMT. And so the clinical findings were that there were an 80% reduction in the overall decolonization post-FMT. There was lower recurrent MDRO infections in the FMT group versus controls, and also versus a group of matched controls. And there was a similar safety profile to those that received the enema. Specifically, unfortunately, I will say that as you saw in the previous slide, the number is small because the trial got started and then COVID happened in the middle of the trial. And so the trial had to be aborted because of the pandemic. Here, this is the Kaplan-Meier curve for decolonization. And so FMT-treated patients had a shorter time to negative stool versus the controls. And this is important because people can MDRO colonization isn't a forever state. And so people can decolonize sort of spontaneously. And so it's important to show, so you see here that the observation group, some of them did decolonize, but this was hastened by the receipt of FMT. And then because our numbers were small, we took our sort of FMT group and we then looked at a group of matched renal transplant patients. And these were about 3,000, or sorry, not 3,000. These were taken from a cohort of 3,000 patients, but ultimately were 16 overall. And you can see these patients you can see there was a longer time to experiencing a recurrent MDRO infection in the FMT treated group. So in summary, so just to kind of summarize this section of the talk, GI microbial communities are critical to MDRO colonization resistance. MDRO colonization frequently precedes infection, and the restoration of the normal microbiome can lead to a reduction in AMR genes, a reduction in MDRO colonization, but beyond that, a reduction in recurrent MDRO and non-MDRO infections, and may lead to reduced mortality. So with this, I'll kind of move on to the next section of the talk. And specifically, we'll be talking about maybe what are some of the challenges of FMT and maybe what are some of the alternatives. So one of the big challenges of FMT, or the big issues with FMT, was highlighted in 2019. And there was 2 NEGM reports where patients who received FMT in a clinical trial setting then had a ESBL E. coli infection. And these ESBL E. coli were then linked to the same aliquot of stool by genomic and metagenomic sequencing. And this actually led to a fatality. And because of this, the FDA issued a safety alert, and this has then led to sort of increase and robust efforts to screen patients or screen stool of healthy donors, specifically for MDROs. And this also has changed the donor eligibility criteria. And so now, if you work in healthcare in any sort of capacity, you are not eligible to be an FMT donor. And so locally, at least for us at Emory, this really made producing FMT and getting stool much more challenging, because we used to have some stool donors that were in healthcare but did not have patient care activities. So specifically, we used to approach the pathologists, and they were always sort of willing and able and generally quite healthy, so did not have any sort of contraindications for stool donation, but this group has now been excluded because of the safety alert by the FDA. And I think another big challenge is how do we know that the FMT was successful? And so what has been suggested as a marker or a measure of FMT success is engraftment. So engraftment is the persistence of donor-derived bacterial strains, and it's thought to be responsible for the sustained improvement in the host dysbiotic state. Specifically, one group have even made a metric called the proportional engraftment of donor strains. And so in this metric, they looked at the number of donor strains that were detected in the recipient after receiving FMT, and then they divided that by the total number of strains that were isolated from the donor. And they found this metric specifically was predictive of FMT success and predictive of FMT long-term success. And in those that had a sort of a high PEDS, or a proportional engraftment of donor metric, they were less likely to have recurrent C. diff many years down the line. And so, clearly there's still a big gap. We still don't have a lot of long-term clinical data and long-term sort of bacterial strain dynamic follow-up data from patients treated with FMT specifically for MDRO decolonization. There is some data within the recurrent C. diff cohorts, but these are a slightly different dysbiotic cohort. So we probably cannot specifically then extract that data to MDRO colonized patients. And so what are some hypothesized mediators of engraftment or hypothesized mediators of FMT success? They span sort of three big categories. One are patient factors. So different patients who are being explored for FMT or may have different disease states. So, and those baseline microbiome configurations are different. So, recurrent C. diff infection patients are different from those that have metabolic syndrome, which are another group that are being treated with FMT. And they're different from IBD patients, which are another group that are being treated with FMT. And they're different from MDRO colonized patients. And then specifically the baseline presence of butyrogenic strains. So as I mentioned earlier, the role of short-chain fatty acids seem to be quite important. And so what your baseline level of short-chain fatty acid production seems to be an important predictor. And then diet has a big role in your microbiome. And so, what is the specific patient's diet and their immune status also plays a role. Donor factors are important. So what is the donor stool? So just because the donor is quote unquote healthy or may pass the screening in terms of their MDRO colonization and maybe the presence of GI pathogens, that doesn't necessarily mean that they have a specifically healthy microbiome. And even what is a healthy microbiome is still quite disputed because there's a lot of heterogeneity depending on where people live, their age, their diet, even if they live in a rural versus an urban environment. And then an interesting sort of factor that is being explored more is donor-patient compatibility. And there seems to be something at play here, but still I think needs to be fleshed out in the literature. Lastly, there are administration factors that are important. So what is the administration route? So giving capsules versus enemas, pre-treatment with antibiotics, which isn't done in, for example, in recurrent C. diff infections or in MDRO colonization, but is done in some other conditions. Patients get pre-treated with, for example, vancomycin and neostigmine, and this is thought to kind of more so wipe out the microbiome, so then the microbiome may be more receptive to receiving new microbiota through FMT. And repeat dosing is being looked at, especially in MDRO colonization. So there's a few sort of industry trials that are ongoing, and they are quite interested in repeat dosing. So, do you get FMT just once? Do you get FMT twice? Should you get 3 days of FMT followed by 3 days of FMT in a month? And so I think there's going to be some sort of interesting data coming out of these studies soon. Okay, so I think we've talked a lot about FMT, and we've talked about what it is. And just to kind of summarize, it's the transfer of feces or complex communities, which can be derived in vitro. And some of the advantages are whenever we do FMT, we're really transferring an intact community. So we're transferring a whole ecosystem. Um, and this can have some advantages because we know that there's probably still a lot that we don't understand. And so we don't want to sort of miss one specific beneficial aspect of the ecosystem. And of course, I think the big advantage is that we have proved efficacy time and time again in clinical studies. So we've proven safety, we've proven tolerability, and we've proven efficacy. Some of the existing challenges, I think, are still in screening of the donor samples. So I think those two NEDM reports of ESBL E. coli transmission still highlight that there can be issues with transmission of pathogens. And I'm sure our transplant colleagues can attest that no matter how much you try, there's still going to be donor-derived infections that happen. Scalability is still a big issue. There used to be one really big donor bank called what was it called? I think it was called Open Microbiome. And if y'all remember, one of our colleagues was working with them up in Boston, who was also a PEDS ID um, uh, a PEDS ID doctor, and now, of course, Mejdi, yes. So Mejdi used to work at Open Biome. That's what they were called. Open Biome have since downscaled tremendously, and so this has caused a lot of issues for centers that were using Open Biome products, and they've had to now maybe start manufacturing their own FMT doses, which can be quite challenging. And of course, there can be some potential variability, as we talked about, in the efficacy, depending on who the donor is. And this is still not well understood. And so this, I think, is still a remaining challenge. So what are some microbiome therapeutics beyond FMT? So I'll touch on at least 4 different classifications: diet and prebiotics, symbiotic microbial consortia, engineered symbiotic bacteria, and microbiota-derived proteins and metabolites. And this is still, I think, a very emerging space. And so we'll only touch on these briefly, just as a sort of introduction. I think the group that I'm most excited about, and I think that are definitely closer to prime time, and you may start seeing these used in clinical practice if you haven't already seen them used in clinical practice, is the microbial consortia. And so precision engineering of a gut microbiota surrogate with bacterial isolates in pure culture can offer the same therapeutic potential of FMT, but without the risks that are associated with the use of raw fecal material. And so this is specifically the transfer of a group of isolates which have been pre-selected or even designed because of specific functions that they're able to restore to the microbiota. So the advantages of using a specific microbial consortia is we know what's in there. We know what we're giving to the patients. We know the specific bacteria that are in there. Unlike in FMT or in feces, you may have a general sort of sense of what's in there. You do 16S or metagenomic sequencing, and you can bin your genomes into maybe species level, or usually taxa is the most resolution that you can get. But here we know specifically what strain we're giving our patient. And so this makes this, from a safety aspect, this is much safer. and we can screen for safety much easier. The challenges, of course, become, which isolates do we select? How do we screen isolates? And then how do we cultivate these isolates? We then need to be able to culture them out. And just because we know what we're giving patients, that doesn't mean that we know exactly what then might happen. Because once you take these strains and you put them into a patient, there's going to be ecological interactions that happen, which again, I think we're still trying to understand. And so just sort of an example, I think a nice study that sort of highlights how these are selected or engineered. So this is a study, it's a mouse study, as you can see, and they took control mice and they then treated them with clindamycin. And then they then had a sort of kind of clindamycin treated mouse model. And then they then administered C. diff 0, C. diff 027B1, which is a particularly virulent form of C. diff, and created a C. diff superspreader mouse. So we have three groups. We have our control mouse, we have our clindamycin treated mouse, and then we have our C. diff superspreader mouse, which is a clindamycin treated mouse or that has also been administered C. diff. And then they took stool from these mice and they looked at the microbiota composition and they found a decreased Shannon diversity index. So Shannon diversity index is just a metric that we use to look at microbiota composition, and it tells us how much diversity of strains there are in the microbiota. And they found in the C. diff treated mice and in the clindamycin treated mice compared to the control mice, there was domination of microbiota with facultative anaerobes. So these are what we typically think of are pathogens or not our healthy microbiota anaerobes that we like to see. So Enterobacterales and Enterococci. And they also found that there was decreased short-chain fatty acid levels in the stool of the clindamycin-treated mice and the C. diff superspreader mice compared to our control mice. So again, sort of getting at the role of short-chain fatty acid production and the role that it plays in colonization resistance. They then took their FMT, they then took their C. diff mouse and treated it with FMT and restored the microbiome back to a healthy control state that looked like the control mice. But then also what they did is that they took a diverse collection of 18 bacterial species from that FMT. So they took that stool that they had specifically used, and then they isolated 18 different species and made them into different mixes. And then they then took the C. diff treated mice and treated them with some mixes, and they found one mix in particular, which they called mix B, cleared the C. diff infection and restored the microbiome back to a control state. And this mix B had some Bacteroides, had a novel Bacteroides species, had a Lactobacillus reuteri, had an Enterococcus hurei, had some novel Anaeropistapes species, had a Staphylococcus species, and an Enteroheptus species, which is another anaerobe. And so I think this is a nice kind of proof of concept of this idea of um, moving from FMT to a bacterial consortia. But we've moved past mouse studies, and maybe some of y'all will be familiar with this 2022 NEGM article, which looked at SER109, which is an oral microbiome therapy for recurrent C. diff infection. So this was a phase 3 double-blind placebo-controlled trial that assessed the efficacy of SER or SER109, which is an investigational oral microbiome therapeutic, which is made up of live purified Firmicutes bacterial spores. And you may be thinking to yourself, why are you saying just Firmicutes spores? What's exactly in there? Of course, this is a proprietary product. So all we know is that it's some sort of Firmicutes consortia or spores of the Firmicutes consortia. And the hypothesis was that these Firmicutes spores would then limit C. diff spore germination, um, because they would compete they would compete for nutritional and metabolic requirements for the C. diff, and also through modulation of bile acid production. And you can see here at 8 weeks, the product, CER109, was superior to placebo. You can see there was a 12% recurrence versus 14% recurrence rate in this group. And so I think this is a nice, again, a nice next step in the production and the study of these microbiome consortia. And I would suspect that many of you, especially people who practice here in the States, are going to start seeing this product being used in clinical practice and GI outpatient practices quite soon. So what are some of the other microbiome-based therapeutics? So engineered symbiotic bacteria. So this is a transfer of a group of isolates that have been designed and selected and engineered to promote specific microbiota functions. So the difference between this and the the consortium, is not only are you transferring select isolates, but these isolates have been genetically engineered or genetically manipulated, for example, through use of CRISPR-Cas9, to in a way that is thought to maybe be restorative to the microbiome. So there's been sort of some E. coli and some lactococcus isolates that have been used as delivery of recombinant proteins, and so this is kind of, I think, still an emerging space, but quite exciting. So the advantages are, again, what you're giving, what the strain is, and you can specifically design, let's say you want to specifically design a Bacteroides species that is a short-chain fatty acid super producer, and you want to sort of restore short-chain fatty acid production to a normal level. I think some challenges, of course, are still figuring out how to do this, um, how to manipulate, which species are best to manipulate, which species would be the best vehicle for this, and safety in humans has yet to be demonstrated. So I think this is still um, something that's still in kind of hypothesis stage. Um, So next is diet and prebiotics. And so I think this is another really interesting avenue. And so this refers to the supplementation of microbiota targeted substrates to promote desired compositional changes in the microbiota. So this is quite interesting because we know that diet has quite a big impact on the microbiome, and there are certain studies that look at microbiome composition shifts with different diets. And so, for example, a plant-based diet has been found to increase prevalence of Firmicutes members that metabolize fiber and complex polysaccharides. And so it's really the body responding to what you're putting into it. So if you're eating more fiber, then the body produces bacteria, or there's a compositional shift in the bacteria that need to break down this fiber. Um, and animal-based diets have been found to lead to an increased frequency of Bacteroides and other bile-resistant symbionts. And I think where this is kind of most promising is in the treatment of childhood malnutrition. So childhood malnutrition has been treated with FMT and has been found to be successfully treated with FMT. Um, and so in this space, there have been microbiota-directed complementary foods, which have been produced and generated and tested, and they've been found to demonstrate an increase in the abundance of bacterial species associated with normal microbiota maturation. And so, of course, the advantages here is that relatively this is easier, so easier to prepare, and of course it's safe. I think the challenges are still that we're still looking at certain outcomes. What's the durability? So if you change your diet, do you then revert back? And for diet-targeted therapy to work, there have there have to be target species already present to then act on the supplementation or the food products that you are ingesting. I think the last group is probably the vaguest group, the most kind of pie in the sky group, and this is microbiota derived proteins and metabolites. So these are the metabolites that we think are mediating the positive response. And so these include things, again, like short chain fatty acids um, as a potential therapeutic avenue. Um, so the administration of short chain fatty acids is being explored. Um, and there's some kind of theories that maybe we can give short chain fatty acid supplements or short chain fatty acid products after the administration of antibiotics. And I think a second big category are secondary bile acids or primary bile acids. Again, these clearly play a role in colonization resistance, and so they're being explored as supplementational therapy or primary therapy. And so again, I think the advantages are these can be easier to prepare. Um, it's easier to assess their safety. And here again, and maybe I should have said this in the diet, these should be safe for immunocompromised patients because we're not giving any sort of live bacterial therapeutics. The challenges, I think, are still, trying to determine the best routes of administration. So, what is the adequate concentration? And then how should this be delivered? Should it be delivered de novo? Should it be delivered in conjugation, maybe with bacterial products? I think it's still a little unknown. So, kind of in summary of this portion, FMT still has many logistical challenges, um, and alternative microbiome therapeutics have promises, but need further study. But I think we're getting to the next stage of bacterial consortia therapy. So I would be on the lookout for some of those trials and some of that data, because a lot of those are in phase 2, phase 3 studies at the moment. So I'll stop there and I'll say, thank you very much for everybody's attention and for listening. And I'll take any questions that I can hopefully answer.
Harry can go on. Let's see that raising his hand.
A nice comment by Nidal. Actually, that's very interesting you say that. So there is a lot of interest and study of fermentation, fermented foods. Um, and so these include things like, asida or injera, for example, in Ethiopia, or in communities that maybe eat more yogurt, or in communities that use fermented foods such as kimchi in some Asian communities. And so, yeah, this definitely, I think, something that should be looked at. Yes, Yusri, I see you have your hand up. Please go ahead.
Hey Ahmed, Assalamu alaikum.
Alaikum assalam.
Long time. Thank you so much. It's fantastic talk. Thank you so much. Ahmed, we've got these multiplex GI multiplex PCRs that are detecting
Mhm,
all MDROs now, of course, in practice in hospital,
mhm,
and we've got the GI physicians who are trying to chase every bacteria that is detected by this multiplex, trying to treat it, uh,
mhm,
blaming it for the cause of whatever IBD the patient has.
mhm,
But I've also heard you talking about pre-treatment as a kind of an adjunct that may make a transplant stick. Uh,
mhm.
so, what is your take on this? So, do we go for that because that is so to use an antibiotic to forget about the C. diff, other MDROs to treat it first before we try to do something? What is the thinking now about this?
Yeah, great question, Yusri. And yeah, we clearly have similar problems with some of our physicians here trying to do the same sort of things with the rollout of these multiplex panels. So I think the GI patients or the GI literature is what's really sort of looking at this the hardest. And some of the trials, they are setting up their trials where they are doing a pre-treatment with specifically with like GI decontamination regimens. So kind of like the old school. I think maybe in Europe you guys use them more, but like the neostigmine and even PO vancomycin. But this is done in quick succession with the FMT. So usually they'll pre-treat. Maybe they'll do like a 5-day course or a 3-day course, and then they'll give the FMT immediately after. And the thought behind this is that it could increase engraftment of the donor strains in the donor stool. And there is, I think the data, there is some, I think, positive data there. And so I'll be interested to see how this pans out maybe in some of the MDRO colonized patients, because I also have seen some prelim data from some industry studies, and some of them are including this in their study design. But I think pre-treatment without a plan for rapid FMT administration wouldn't really make sense to me. I think Hamid, you have your hand up, and I see Zahir's question, which I'll be happy to answer after Hamid comments.
Thank you, Ahmed, for your presentation. As you already mentioned, this is an important area. I remember actually when people were talking about microbiome years ago, we were very dubious about its role. I similarly attended a lecture actually in the role of microbiome in cancer risk and cancer therapy. Again, very exciting. So my question is about resistance and virulence. A lot of sort of studies in resistance show there is no direct correlation between resistance and virulence. And I'm very interested in the Scandinavian study, as you mentioned, patients who are colonized with MDROs went to have invasive disease evidence by bloodstream infection or bacteremia. So what is your comment about colonization, about MDROs and acquiring of virulence factors? Yeah. Thank you.
Yeah, I think that's a great point and a great question. And I think at least in a lot of the studies that I've been sort of reading and consuming, the focus is always on resistance and not so much in virulence. And I think the next stage, and maybe I'll kind of loop in Zahir's question here. I think a lot of people are now interested in looking at how different gene pathways or gene or sort of metabolic, like what is the change in the gene content of the microbiome, and what's easiest to look at is AMR gene content, because we have very well curated AMR gene databases that are updated, that are maintained, that you have 3 or 4 or 5 different groups at a time that have AMR, and so it's I think it's easier also to look at that. There are some virulence gene databases, but I don't think that they are well characterized, or some of them to me sometimes seem a little hand wavy. They'll have stuff like heavy metal resistance, which we know can play some sort of virulence role, but maybe those just play a role in persistence in the environment. So I think it's still quite unknown. And I think to your point is that in is subsequent infection, is that a role? So if you're colonized with an isolate, is subsequent infection or subsequent invasive infection a role of virulence or a role of resistance? And it's probably hard to say, but I do think that some of the data shows that there's even a reduction in non-MDRO infections. And so maybe there is something at play from a virulence standpoint, or maybe just from an overall health immune standpoint. So I think that's my take, but I think clearly I also I'm still learning about some of this. Okay, so to answer Zahid's question, so Zahid, typically what people look at for microbiome composition, or is that still on? I don't know if that's still on, but I'll just answer for the group anyway, is we look at what's called alpha diversity and beta diversity. So alpha diversity is the diversity of your species. So I think a nice example would be if you look in a room, what's the diversity of the people in that room? That's alpha diversity. Beta diversity is comparing different groups to each other. So if you look in two rooms, how similar are those two rooms to each other in terms of their diversity? That's beta diversity. So the main metric is alpha diversity, and there's different mathematical metrics such as like Shannon diversity, which people look at. I think really what people tend to do is they tend to look for reversion of the microbiome, maybe. So increase in alpha diversity is a really easy one. Does it then start to mirror the donor microbiome is something some people look at. But then looking at the reduction of your facultative anaerobes. So looking at reduction of your Enterobacterialis and your Enterococci, for example, and an increase in some of the anaerobic taxa. We have some data in some of our studies where we have identified particular anaerobic taxa and species that we think are maybe good markers of of healthy microbiome post FMT, and these are things like Phascolarcobacterium and some other species. So, and I think I tried to highlight that engraftment question because I think that's still very unknown. So people are also looking at this theory of engraftment. So do we see the donor strains in the stool, and how long do we see them in the stool? Do we see them for a few days, for a month, for a year? Is that what's important, that the donor strains that they persist, or is it just important that there's some sort of compositional shift? I think it's still a pretty unknown question. But of course, the most important thing is the clinical condition. So in recurrent C. diff, I think that's an easier question to answer. So if you don't recur, then it's been successful. But unclear in some of the other disease states. Yes, Zahid, I do. Zahid, what I envision, to be honest with you, is I envision that we may get to a place where there's potential co-administration of antibiotics and maybe some sort of probiotic, or following antibiotic administration, some sort of microbiome restoration therapy that's given. There's interesting data in the BMT literature that is looking at banking of patient stool and then giving them their stool back after their BMTs to prevent graft versus host disease. And so there could be a world where, let's say, you are about to receive antibiotics, or you are about to receive chemotherapy, or you're about to undergo some major, maybe something that we think is going to majorly alter your microbiome. Maybe you're going to submit some stool, and that can be banked and refined and it given back to you. Or maybe we're going to be in a place where we're going to have a small microbial consortium maybe 2, 3 bacteria that are given, and they're co-formulated with maybe some kind of short chain fatty acid product. So that's kind of where I envision it. I do think because of scalability and safety, we're probably going to move away from fecal transplant and move into consortia. Yes, Azadine, go ahead.
Thank you, Ahmed, for the nice presentation. I'm circling back actually to Nidal comment about Sudanese Asila and Kisra. And just my question is, with all we know here in the US and the Western Hemisphere, we struggle with the recurrent infections and resistance and C. diff. But what about resource limited countries? Is this a problem? Is this any research similarly done with the fecal microbiota there? And the other question is also, anyone look at using fecal transplant from areas from like Africa, resource limited countries, any other totally different population, and compared to us here, or use it in our patients in the US? Is that something somebody looked at? Or
Yeah, I think that's a great point and a great question. And I think it all stems back from, so there's definitely observational data or sort of survey type data that the microbiome differs and it differs in different parts of the world. But even there's one study that I once read that even in, I think it was in Ecuador, and even if you went from the capital of Ecuador to like a village or to a very rural population, those microbiome um, signatures looked different, and those microbiomes looked more similar to each other. And so I think it's an interesting question. I think the safety aspect is always going to be an issue, and I think probably like the FDA would balk at us proposing bringing a stool from outside the U.S. To try and use it. But where it has been studied, as I had kind of previously mentioned, is in malnutrition. And so some of the malnutrition data is in Africa. Um, and our group is quite interested in exploring FMT in resource limited areas. And so we've started some studies where we're going to look at acceptability amongst, we have a small pilot grant in Ethiopia to look at acceptability of FMT. So if FMT was available, how would patients respond to that? How would providers respond to that? Would they be open to this? Or would there be other social barriers? But I think specifically to answer your question, I'm not aware of any specific studies that have looked at, is the microbiome composition in other countries, and specifically maybe in low middle income countries where the diet is usually more plant-based, may have more fermented foods. Could that be better? I think, I'm more familiar with the MDRO literature, FMT for MDRO. So I'm not aware of anything in that literature, but FMT is definitely being looked at for a lot of different conditions. One that comes to mind that maybe they're looking at this for is obesity. There is a lot of interest in FMT for obesity. And I think there was a nice little anecdotal case report a few years ago from the ID literature where I think a daughter was an FMT donor for her mother for C. diff. And there was quite a change in the mother's body habitus, which was attributed to her receiving FMT from her daughter, and her body habitus then changing to sort of mirror her daughter's after FMT. It was just like one anecdotal case report, but it was a kind of an interesting report.
All right. If there's no questions, we thank you, Ahmed, for the very interesting and nice presentation. And thank for everybody who attended. And we'll see you again next month.